Did you know that viruses are not living organisms? When you were vaccinated against COVID-19, you were injected with modified viruses. Viruses cannot reproduce on their own. They can only be created by living organisms, including being produced by your body. About 8% of your own genetic information is brought to you by viruses. If you didn't know this, I invite you to watch this video. I will try not to be boring. My cat Martin will make sure of that. So, since childhood we have heard the word microorganisms without really understanding what it actually is, and they can be very different. Let's quickly look at a few types. Bacteria are tiny single-celled pests. We, anthropocentrically, divide them into harmful, harmless, and beneficial. Harmful ones mercilessly gnaw at us, sometimes wiping out large chunks of the human population. These are well known to humanity as plague, cholera, tuberculosis, various food infections, and many others. Bacteria are single-celled. That is, they are simple living organisms with a membrane that can protect them, mask them from the immune system, and help them stick to surfaces. And the filling consists of different parts, like a microfactory for protein synthesis, an area where the bacteria's DNA is located, and other components. Additionally, there might be a tail that allows the bacterium to move. The main point is that it is a separate organism. There are also beneficial bacteria. They have long lived in symbiosis with humans, and without their help, we would struggle. Beneficial bacteria mainly live in the intestines and digest what we cannot digest. Some bacteria even produce the vitamins we need. Moreover, they often protect us from pathogenic bacteria. Bacteria reproduce by division. This is when one organism divides into two smaller ones. In addition, many other bacteria in nature do not affect our bodies. They also process some substances into others, contributing to the natural cycle of substances. The next big group is fungi. This is a large group of organisms, but let's quickly look at only single-celled ones. The largest group of single-celled fungi is the yeast we love. Everyone appreciates their work. Some love buns, others bread, and some enjoy beer with whiskey, the best friends of the modern glutton. There are also not-so-pleasant yeasts, such as those that cause candidiasis or dandruff. But in nature, all are equal. The structure of yeast differs significantly from that of bacteria. Single-celled yeasts even have some semblance of a skeleton and a more complex DNA organization. Yeast reproduces by budding. Some yeasts even have a semblance of sexual reproduction. As for whether yeasts have a marriage institution or reproduce in sin, science knows nothing. Moreover, even religious organizations are unaware, and as we know, they can explain everything in the world. Before moving on to the exciting description of viruses, we need to briefly clarify what genes, DNA, RNA, and ribosomes are. Ribosomes in cells are factories of necessary parts. They can produce various chemical compounds, but they need the necessary instruction on what to produce. DNA, deoxyribonucleic acid, is a complex large molecule. We have all seen its image in various movies or articles. Essentially, DNA is like a flash drive that stores all the information about a creature, and such a molecule is in every cell of a living organism. A gene is a part of a DNA molecule that is responsible for a specific function or trait. For example, there are genes responsible for eye color or predisposition to obesity. Genes contain instructions for creating proteins in ribosomes. Proteins perform many different tasks in the body. They can be building blocks for cells or perform various functions in tissues, such as aiding in chemical processes like digestion. RNA, ribonucleic acid, is a molecule that transfers information from DNA to ribosomes. RNA copies the necessary instruction from DNA and delivers it to the ribosomes for production. Most living organism cells have DNA, RNA, and ribosomes. By the way, an interesting fact. Although the calculation is not very accurate, approximately 750 megabytes of information are recorded in DNA. That is the volume of a data CD. This was a brief theoretical excursion to understand that viruses are something fundamentally different and unusual. So, viruses. When I was preparing the material, I came across such a definition in some places. Viruses are microscopic infectious agents. I don't know who came up with this phrase first, but they are clearly an enemy agent. What can you even understand from this phrase? I asked DAL to generate an image of this description. Here are the agents. Let's try another way. Simply put, a virus is a foreign genome, DNA or RNA, covered by a protective shell. Plus, the virus has a way of penetrating a cell. For different penetration options, viruses take on different forms. Probably, people around the world know what the coronavirus looks like. A spiky ball. The virus's spikes are made of a material that clings to specific receptors found in some types of cells, particularly in the lungs. The cell, sensing something has stuck to the receptor, assumes it is some nutrients and absorbs the virus. A slightly different penetration mechanism is used by spiral-shaped viruses. Such a shape is found in flu or rabies viruses. They also stick to the cells they need, but their shell structure allows them to literally fuse with the host cell, like soap bubbles merging. But bacteriophages take first place. The victims of these viruses are not the cells of living organisms, but bacteria. It's harder for viruses to deal with bacteria. Bacteria's skin, or shell, is tougher than cells, and it's more problematic to penetrate them. But not for viruses. The structure of a bacteriophage resembles a syringe or a drilling machine. It has legs that attach to the bacteria, a tube with a needle and a spring. After attachment, the spring pushes the needle through the bacterial shell, and then the genetic material from the virus head is injected into the bacterium. Can you imagine that? And remember, the virus is not alive. It's just a mechanism that copies itself. I was shocked when I found out. Even Cat Martin hiccuped and didn't finish his sausage when he heard this, although it seems he just fell asleep. But that's not all about viruses. We remember that the virus genome is in a kind of protective capsule. After the virus enters the cell, it undresses, meaning it sheds its capsule. The capsule is made of materials that the cell's internal environment dissolves, releasing the virus genome. So, the virus genome enters the cell, and if it's a DNA virus, it replaces one of the human DNA genes in the cell. Then the human cell's RNA will start carrying this foreign information to the ribosomes, which will produce parts for creating new viruses. If the virus carries RNA, the process is even simpler. It doesn't even have to replace a cell gene. The virus's RNA enters the cell and is perceived by ribosomes as an instruction to assemble proteins. Accordingly, they again produce parts for new viruses. An example of an RNA virus is the well-known COVID-19. Next comes the process of assembling viruses from parts. Describing this process simply is difficult. It is complex and convoluted. Let's leave it aside. But remember that it is not robotic arms or spiritual energy that assemble it, but processes or algorithms described in the same viral genome. So with songs and jokes, new full-fledged viruses appear. Sometimes they tear the cell apart, killing it. Sometimes they only damage part of the membrane, bursting out. But after that, the flash drives are free and rush to infect new horizons. So in a nutshell, that's how viruses reproduce. It somewhat resembles the reproduction method of some parasites in nature. Except parasites have a reason to do it. Parasites are living creatures, and this is their way to survive. But what benefit does a virus get from this? It is a kind of thing in itself. Of course, everything I'm telling is just a general, very simplified description of all processes for a rough understanding of what is happening. It is not recommended to apply to medical school with such preparation. But let's return to who ordered all this music. Here are some theories of virus origin. Coevolution theory, or evergreen theory. It is believed that viruses appeared long ago, even before the first cells. They could have evolved alongside cells, constantly adapting and changing. This theory suggests that viruses always existed as separate agents and evolved alongside other forms of life. I must say that I haven't delved deeply into the theory, but saying that mechanisms of cell impact appeared before the cell itself is somewhat strange. Degradation theory or reduction theory. According to this theory, viruses were once more complex organisms that gradually lost most of their cellular functions, becoming simple infectious particles. They could have originated from cells that lost the ability to survive independently, turning into parasites. This theory is more interesting, but does not explain the diversity of viruses in nature, and there are many variants of them, both for animals and plants. Mobile genetic elements theory. This theory states that viruses originate from mobile genetic elements, such as plasmids, small rings of DNA that can move between cells. Plasmids could have gained the ability to infect cells, becoming viruses. Again, where does the diversity come from? Hybrid theory. It is believed that viruses could have arisen from a combination of different processes, including mobile genetic elements and degraded cells. This complex mix of mechanisms could have led to the emergence of viruses. Well, those are the theories. I'm not a biologist, so I have the right to my own theory, even if it sounds silly. My theory is based on the property of viruses to transfer genetic information. Viruses handle genetic information quite casually and can change and modify it in the following ways. Mutations. Viruses can mutate, meaning their genetic information can change due to errors when copying the genome. This is especially characteristic of RNA viruses since their copying mechanisms have a high error probability. Recombination. Viruses can exchange genetic information with each other if they infect the same cell. This can lead to the creation of new viral strains with a combination of genes from two different viruses. Reassortment. For some viruses like the flu, genome segments can be transferred between different viruses during simultaneous cell infection. This can quickly create new virus variants. So viruses have different ways to change and update their genetic material, allowing them to constantly adapt and survive in various conditions. These processes also make it difficult to fight viruses, as new strains can be resistant to existing vaccines and drugs. Additionally, some viruses, especially retroviruses like HIV, can integrate their DNA into the host's genome. This means that the viral DNA becomes part of the host cell's genetic material and can be passed on to subsequent generations of cells. Host information in virus genomes. Viruses can capture host genes during the infection process and incorporate them into their genome. This can happen during recombination or other genetic exchange processes. Captured genes can change the virus's properties or help it evade the host's immune response. Scientists have discovered that 8% of the human genome is remnants of viral genomes. Can you imagine the scale? So maybe viruses are not a problem, but a blessing? No, not for us as individuals. It is hard to call polio, HIV, or hepatitis a blessing. But for the population as a whole, I don't know who first called nature mother, but that mother cuts down individual organisms so ruthlessly that it's a sight to behold. And overall, humans are large and clumsy organisms whose genetic variety selection takes a long time. For development or selection, changes need to be made and the results observed. We receive a genetic set at birth, and we can share it with children only after many years. But our genetic sets are not much different from those of people living around us. But viruses can bring rapid changes to individual genetic sets. This promotes greater variability and accordingly, higher chances of population survival during changes, for example, climatic. Still, those viral 8% of our DNA say something. What if the species that were more susceptible to viruses were the ones that survived in nature? And that's why now probably every living cell can find a virus. Where did viruses come from? Well, if they infect cells, and in a way, have information about those cells, then perhaps they are originally a product of mutated cells. Take cancer cells. They also produce who knows what, but mutated native cells of the body initially generated them. So who knows if we didn't create viruses? Or maybe we live thanks to viruses. But these are all theories, and money doesn't flow under a lying theory. Scientists have long assessed the prospects of using viruses and tried to tame them, and they have been quite successful. Viruses are used in various fields of science and medicine. For example, in gene therapy, viruses are used to deliver healthy genes into the cells of patients with genetic diseases. Viruses are also used to create vaccines, such as against flu or COVID-19, where weakened or inactivated viruses are used to stimulate an immune response. So viruses can be both destroyers and useful tools, depending on how they are used. They are an integral part of the natural world and continue to play an important role in biological processes and scientific research. Viruses are used to control pests. For example, in agriculture, they are used as an effective and environmentally friendly method to control pests. In India and China, the polyhedrosis virus of the mulberry silkworm is used to reduce the population of the mulberry silkworm on plantations. This reduces the use of chemical pesticides and protects the environment. The cabbage moth polyhedrosis virus is used in the USA, Canada, and Europe to control the cabbage moth on cabbage fields. This helps reduce crop losses and improve product quality without using chemical insecticides. In Canada, the pine looper polyhedrosis virus is used to control the pine looper population, which damages coniferous forests. Viruses are used in gene therapy to deliver genetic material into patients' cells, allowing the treatment of genetic diseases. Gene therapy involves introducing healthy copies of genes into cells to correct or replace defective genes. Drugs have already been created and approved to treat spinal muscular atrophy, some types of congenital blindness, or severe forms of hemophilia. These are just a few examples, and the field itself has enormous prospects. Viruses are also used to prepare animal organs for transplantation to humans. For example, in May 2024, a pig liver was transplanted to a 71-year-old patient in China. Usually, transplanted pig organs are often rejected by the patient's body, leading to death. But if the DNA of liver cells is modified, the chances significantly increase. Artificial viruses also carry out these DNA changes in animals. They introduce human genes into the tissue of the organ to be transplanted. Oncolytic viruses are widely studied. These are specially designed viruses that infect and destroy cancer cells. The first commercially available oncolytic virus, TVEC, was approved in the United States in 2015 for melanoma treatment. TVEC, a modified herpes virus, not only destroys cancer cells but also stimulates the immune response, helping fight the tumor. Other examples include the rheolacin virus, currently undergoing clinical trials for treating various types of cancer, such as head and neck cancer. Rheolacin uses the rheovirus, which naturally infects and kills cancer cells without harming normal cells. Oncolytic viruses are also being investigated for treating other cancers, such as lung and colon cancer. The Pexavec virus, based on the vaccinia virus, is undergoing clinical trials for hepatocellular carcinoma treatment. Thanks to viruses, we may finally get those long-awaited treatments for various types of cancer. What about obtaining various drugs? Viruses have found their place here as well. For example, bacteria are ready-made factories for producing the necessary substances. They only need to be asked correctly, and modified viruses know how to do this. One of the most famous examples is insulin production. In the 80s, Genentech and Eli Lilly developed a method for producing human insulin using genetically modified bacteria into which insulin genes were introduced using viruses. This approach allowed the production of insulin in large quantities for diabetes treatment, which was a revolutionary step in medicine. Another interesting fact: did you know how your COVID-19 vaccine works? For example, AstraZeneca and Johnson & Johnson vaccines use modified adenoviruses to deliver genetic material into human cells. That is, you were injected with a modified virus that penetrated some of your cells and forced them to produce the proteins that make up the spikes of the COVID-19 virus. Obviously, your body didn't like such jokes and developed immunity to these proteins. And when someone coughed COVID-19 on you after that, your body was already ready to identify and fight the problem. But viruses are used not only in medicine. Viruses are used to create new materials due to their ability to self-organize and specifically interact with various molecules. For example, the M13 virus has a long, filamentous shape, making it ideal for creating nanowires. Researchers can genetically modify the virus to bind to metals like gold or palladium. After the virus binds to the metals, nanowires form, which can be used in electronics. Viruses can also be used to create nanocomposites. They provide uniform distribution of nanoparticles in the material, improving its mechanical and electrical properties. For example, adding viruses to polymers can enhance their strength and conductivity. In 2006, researchers from the Massachusetts Institute of Technology used the M13 virus to create nanowires from cobalt oxide and gold. These nanowires are used to create anodes in lithium-ion batteries, improving their efficiency and lifespan. In 2020, scientists from the University of California, Riverside, developed a method for creating high-efficiency catalysts for water splitting using viruses to form platinum nanoparticles on the surface of carbon nanotubes. This allows the creation of efficient and cheaper catalysts for hydrogen production. So as you can see, viruses are not some toothy monster like my cat Martin, but an effective natural evolutionary tool. Extremely efficient and promising. People have only recently started harnessing its power. The technology is promising, and artificial intelligence will not be able to take away the profession of a virologist as soon as it will take away the profession of a programmer. Therefore, note that in the first comment under the video, I have selected for you or your loved ones the most interesting books on viruses that may interest both adults and children. And I am not a biologist, but a programmer. And I can only hopelessly teach Cat Martin various tricks, only increasing his contempt for me. Like if you liked it, dislike if you didn't. Subscribe to the channel, and stay healthy.